Hello everyone. So today we're starting to talk about microscopes. Now we can only talk about these a little bit. We do actually need to use them for something, which I'll talk about in a moment, which obviously we can't do right now. So if you don't completely get everything today, don't worry, because obviously it's supposed to be consolidated. We're actually using it, and I'm at the moment just really optimistic that we could go back in June. So hopefully we'll be able to do this before the end of the year. Right, so title for today's lesson would be The World of the Microscopes. And the first thing I want you to do, it says presenting how you'd like, match the items on the sheet. Now the sheet I have attached, and it's this one from Publisher. Now obviously you guys haven't got the diagram, and I can print this off for you next lesson when I actually see you, that is. But all I want you to do is basically match all these different parts of the microscope to their function. So I have put the answers on the slide. So the answers are at the bottom of the PowerPoint slide down here. I mean, obviously you could cheat, couldn't you? There's no point in cheating. You're only cheating yourself. You actually will learn more if you try and figure it out yourself first, because you're more likely to remember it if you've gone through that brain function. I know that a lot of people don't like to write it down wrong and then have to correct it. So what I would kind of do is you read through it, try and attempt it, maybe you just kind of number them up on a post-it note or something, and then check the answers and write them out in the right order in your book. So you can either choose to pause the video now or do that in a minute, but that's what you need to do. Going on and talking about two different microscopes. So you need to know about two different microscopes and where they come from. So remember, the microscope, the word micro means small, scope means view. You could add that to your glossary. So light microscopes are basically what we have in school. They started to be developed in the 16th century when the first sort of microscope was about. It used to be called the simple microscope because it only had one lens on it. As you can probably see from the diagram that you've just kind of done the fill in on, we have on this one 4 different lenses. You have an eyepiece lens here and objective lenses. So there's 3 objective lenses there, and you can basically choose which one you want to use. And they're all different magnifications. So it used to be called the simple microscope when it only had one lens, and now it's got a minimum of 4. We call it the light microscope. The reason why it's called the light microscope is obviously because light shines through the specimen that you're trying to look at, and that's how you see it. These are the ones we've got in school. If you were to buy one brand new, it would probably cost you about £400. Now we need to compare that to a different kind of microscope called the electron microscope, which is the one you can see here. Now an electron microscope was developed far, far later, which is about the 1930s, and since it's been developed, that's partly how we've learned so much about cell structure. So things that we did last lesson about talking about different organelles in the cell, ribosomes, mitochondria, chloroplasts, vacuoles, etc. We would not know any of that if it wasn't for this electron microscope. We needed to know about the electron microscope in order for us to learn all those things. So if you can see here, a light microscope can magnify up to about 2,000 times the size. The ones in school, I've put 1,000, but I'm not even sure they go 1,000. So to work out the magnification, you do the eyepiece lens, which is normally times 10, times by whatever objective lens you've got. So actually that should be 400. Because in school, the eyepiece lens is always times 10, and then you have 3 lenses, times 4, times 10, and times 40. In order to work out the total magnification, you do the times 10 times by whatever objective lens you use on, so the maximum would be times 400. The maximum a light microscope can zoom in is times 2,000, okay, and it costs about £400, as we said before. An electron microscope, however, can magnify like 2 million times. Okay, so it's much, much more powerful. However, it is much more expensive. Number one, its running costs are much more expensive. The light microscope just needs you to replace the bulb every now and then, look after it, and plug it into an electrical socket. The electron microscope is pressure sensitive and temperature sensitive, and all the conditions around it, like moisture levels, need to be very, very specific. Because of that, it takes more energy and therefore more cost to run to keep and maintain those conditions. It also costs roughly like £1 million to buy. So, you know, we obviously don't have one of those in school. Most universities probably do have one, but only if they have quite a large science department. And the likelihood of that they have more than one is not huge. So they are really expensive pieces of equipment, but you can see so much more. On a light microscope, you'd kind of expect to see cell membrane, nucleus. That would be it. Everything else would be a blur. On a electron microscope, you can see all the individual organelles, and that is the difference. I want you to pause now and have a go at answering this question. Remember, it's 4 marks, so think about how much you need to say. Once you've done that, I want you to have a go at this, thinking about conversions. Okay, again, I've put the answers down below, but have a think, what can you convert to units? Now, I've added 2 more units in, and this, again, and I apologise, this is where it's getting complicated, and it would be nicer if I was there in real life. We have 2 more smaller measurements that you need to learn about. They are called micrometers and nanometers. So if we split 1 millimetre, that tiny millimetre, 1,000 times, you can get, that would be a micrometre. So there's 1,000 micrometers in 1 millimetre. So picture that on a ruler, basically, how small that is. Now in 1 micrometre, we also have 1,000 nanometers. Okay, so massive difference between a nanometer and a kilometre. Like, they are tiny, these are huge. So to give you an idea about why we need to use microscopes to see cells, the average size of a cell in our body, for example a red blood cell, its diameter is about 7 micrometers. So that's why you can't see it with the bare eye, and that's why we need to use microscopes. So have a go at filling that table in. Again, the answers are at the bottom. Now, this is just briefly something I need to kind of introduce you, the idea of a required practical. Now, required practicals basically are practicals that you have to complete during your GCSEs, and you need to know the method of how to do it, what the results should look like, what the different variables are, all those kinds of things. Now, this would have been a required practical, which is why you will absolutely, without a doubt, do it at some point. Hopefully the sooner we return to school, the better. But we would at this stage be doing the required practical, which would involve us actually operating a microscope and learning how it works and its functions, calculating the magnification and the image size and the size of the object, and drawing the image. Now, unfortunately, we can't do that at the moment, but what I can do is sort of talk to you about this stage. So we have a triangle. We call it the I am triangle because it says image, actual, magnification, I am. That's the easiest way to remember the order of it. Okay, it's the only formula triangle you need to know for biology, which makes it better than physics, of course. And it basically means that in any exam question, they're going to give you 2 pieces of information out of these, and you need to be able to work out the third. So they might write an exam question such as, Precious is using the microscope to view a cell. She uses her ruler to measure the image, and it measures as 7 millimeters. She has a magnification of times 50. What is the actual size of that cell? Now, the way I see it is, that sounds quite simple. The image size was 7, and you divide that by the magnification, which was times 50, and then you get your actual size. Quite simple. The thing that makes it more difficult is actually thinking about how we measure them. So we know that magnification is going to be the eyepiece lens times the objective lens. We know that the image size is going to be measured in millimeters, and the actual size is going to be measured in micrometers. Now, the hard thing about that is doing the conversions. So I'm going to record a separate video for you that actually goes through an example of those questions. You need to watch that before you complete the next slide. So the next slide is just giving you a few different tasks. It means you need to draw the triangle in your book and annotate it, say what it means. You need to explain how you'd work out those different things, and then you're going to answer some questions on a worksheet that I'm attached separately. And it's just to kind of practice you doing those calculations. So stop what you're doing, switch to the other video, and we'll go through a question together.